Okay, hi everybody. Thanks for coming out. It's a packed crowd today. That's exciting. So this is one of 2 weeks where our fellow EEPB graduate students are gonna be giving updates on their research, and the idea is just to help the grad students get to know what we're actually up to research-wise, because we know what we're up to in everything else already. So we're very excited to hear from our fellow students and have some friendly feedback. So up first, we have Mahal Buguy. She graduated from Agnes Scott College down in Georgia in 2019 with a BS in biochemistry and molecular biology. She is a current PhD candidate in Rachel Peczkowski's lab. Outside of research, she enjoys watching movies, embroidery, and not falling off her longboard. So give it up for Mahal.
Can you all hear me okay? Fantastic. Please let me know if I'm talking too fast. And I'm just going to get started on my chapter 1 work so far. So starting off really broad, we have the microbiome, right? The assemblages of bacteria and fungal communities. And we're probably really familiar with how the human microbiome, especially the gut microbiome, can provide us insight into many health issues for humans. And much like how the human gut microbiome impacts our health and development, plants also have their own unique microbiomes that can impact their own health and development as well. For instance, microbiomes can help protect against pathogens and help acquire nutrients. And when studying a plant's microbiome, we can see that environmental factors such as soil chemistry can interact with microbiota, which can impact a plant's growth and health. And by studying a plant's microbiome, we can utilize that knowledge and develop applications for agricultural purposes. For instance, we can use microbes to protect plants from pests and to help promote plant growth. And a lot of this research has been done in agricultural systems. However, for my work, I'm interested in applying these same concepts into other systems, such as wild plants in our cities. And much like how studying the human or agricultural plant microbiomes can offer us insight into their respective systems, researching wild plants and their microbiomes in cities can give us an insight into the conditions of our own backyard. And here we have a photo of the St. Louis Arch. Hopefully you all recognize that by now. So why even be interested in cities? According to the United Nations, they expect the global urban population to grow from 54.5% to 60% by 2030. And as you might imagine, to match this growing population, cities are going to increase in their size and development. And with this urban development, we will see increased disruptive and long-lasting modification of the environment, such as increased habitat fragmentation due to more road development. And for my research, I am particularly interested in the introduction of pollutants into the environment, especially soil pollutants. So to be more specific about soil pollutants, I'm interested in heavy metals such as lead and cadmium. We're interested in these heavy metals because it's a way to measure how urbanization affects the natural environment. For those that may not be familiar with heavy metals, they can be naturally found in the soil, but their concentrations can be increased due to human activities such as industrial waste, construction work, leaded gasoline, and paints, as those living in older homes might be familiar with. And heavy metals have a higher retention rate in the soil, meaning that they're difficult to break down, and so they stay in soils for long periods of time. And that creates a public health concern for a lot of reasons. For instance, people living in older homes that have leaded paint, it could be a danger to your child if they consume leaded paint. And if you're near a demolition site that has contaminated soil, you can breathe in the leaded soil as well. And heavy metal exposure can further damage the nervous system and even cause cancer. And so that image over here is a lead remediation project actually in St. Louis. So up until now, I've sort of described the relevancy of cities and heavy metals, but where do microbes really fit in for this research? And so there's a growing number of studies that focus on how microbes respond to urbanization, such as urban stress, changing bacterial composition. Other work has linked increased heavy metal accumulation to decreased soil microbial biomass and microbial activity. However, there isn't much work done connecting the soil chemical properties in the soil with the bacterial and fungal communities of rhizospheres across an urbanization gradient. And by rhizosphere, I mean root-associated soil of plants. And this research gap is really the focus of my work for this chapter. So this leads me to my research question: how do 1, soil abiotic, and 2, soil biotic properties vary across this urbanization gradient? So by soil abiotic, I'm referring to the soil chemistry, such as heavy metals and macronutrients. By soil biotic, I'm referring to the soil microbiome, what bacteria and fungi are present in that soil. And in this project, my focus is on urbanization gradient across the greater St. Louis region in Missouri. And in this map of land cover over here, the really red part, especially in the city here, that's increased urban development, like increased impervious services, et cetera. And as you can see, as you get from the really red city down further into the counties, it's a lot more green, meaning that it's less developed in terms of impervious surface. So for this study, I had two predictions. One being that the soil abiotic properties will vary with the level of urbanization intensity. So urban sites will have higher concentrations of heavy metals compared to the suburban and rural sites. And the second prediction was that the soil microbiomes will also vary with the level of urbanization intensity. So soil microbiomes will be similar within a given site type. So rural sites will be very similar to each other. Urban sites will be very similar to each other, but they will vary between the site types. So I would expect that the urban and the rural sites would be very different from one another. And so going kind of into the study design. So looking at the 10 sites I had across the St. Louis region. So I had 2 rural, which is defined by the percent built environment, 5 suburban and 3 urban. And you can see the distribution across. So the farthest one out is all the way in Shaw Nature Reserve. And I'm going to say this earlier, but yeah, the colors represent the different site types. So rural is green, suburban is purple, and orange is urban, and that's reflected throughout the entire presentation. So now I'm showing a PCA of the different sites and different environmental variables. And the site types are again classified by those same colors. Then the gray vectors of this PCA tell us how each of the measured variables influences the PC axes. And the vectors with a smaller angle between them are positively correlated with one another. So you can see that mean temperature, intersection density, and nighttime radiance are closely together. They have a smaller angle between them, meaning that they're positively correlated with one another. And so really just to summarize, like what to even get from this graph, this PCA is that urban plots have higher density of roads, they have more impervious surfaces, they have less forest cover, warmer temperatures, and lighter night skies. And the point of this PCA is just to verify that yes, not only do these sites differ in percent built environment, but they differ in all these other environmental variables as well. And so going into the focal species of this study, I had 2 Plantago species, Plantago lanceolata and Plantago regelii. These were chosen because they're abundant across the urbanization gradient, which means they make a good model for studying plant microvariation across common species. And if you want to read more about them, well, looky here. Our near and dear Rachel Penchikowski has her own review in the American Naturalist about this family, along with Drew Sig at Truman State. And so going back to what I actually did with these plants, so I I sampled soil from 5 plants of each species at 10 sites, so a total over here of 100 soil samples. And here we have a little cartoon Plantago lanceolata, because you're wondering, what do you even do with these soils? Well, I'm about to show you. So first we have the little soil near the roots, collected some of that soil, and then the analytical biogeochemistry lab at WashU did the analysis for trace metals, heavy metals, and macronutrients. And then for my part, I again collected the same soil from the same plant and froze it, extracted its DNA, and did dual index metabar coding. So 16S was done for the bacterial communities and the ITS was done for the fungal communities. And now, not this step, at a computer. And now I'm gonna transition to some of the preliminary results that I have. So starting with the soil chemistry data, showing again another PCA of each site's mean concentration of several macronutrients, the trace elements, and heavy metals. And again, the gray vectors tell us how each of the measured elements influences the PC axes. So from this plot, we see that the urban sites, the orange dots, have higher cobalt, nickel, which are macronutrients, and higher lead and cadmium, which are heavy metals. The suburban plots, the purple dots have higher cobalt, manganese, and phosphorus, and rural sites, the green dots, have higher sulfur, calcium, magnesium, and silica. And the key takeaway from this PCA biplot is that, yes, the soil chemistry does vary with urbanization, as we can see the different elemental vectors distributed across the different site types. And so now transitioning into more of the microbial data. So we have these two ordination plots, NMDS plots, if you're that interested in knowing about them. And the way to read ordination plots and NMDS plots is samples that are very similar are going to be closer together, and samples that are not as similar are going to be farther apart. And so I have two of these plots for bacterial and the fungal communities. And right now, this is just for the Plantago species as designated with this green square for lanceolata and this orange triangle for Buguy. And so looking at the bacterial and fungal communities, we do see that there is overlap of the microbial communities of these two species, with at least lanceolata seeming to have a broader range, and Buguy having a smaller range that's kind of nested within the lanceolata community. And looking at Adonis tests, so permanovas, to confirm, like, does Plantago species, is that a good predictor, I guess, of the differences between these two groups. And so far it seems that, yes, these do seem to be significant. And now we have, again, two NMDS plots, but this time these are grouped by site type. So again, sites, rural, green, suburban, purple, urban, orange. And again, bacterial and fungal communities. And so you can see that the orange dots for urban here are clustered with each other. Rural is over here, and that suburban is in the middle. You see that urban and rural sites are different from each other. They're not really overlapping. And the suburban sites are kind of in the middle of those. And this is reflected also in the fungal communities. And doing the permanova, the Adonis test confirms that, yeah, site type can explain a good amount of the variation between these site types. And if we were to further split this data of the site types by Plantago species, so it's a little bit much. So the site types still reflected that color, but Plantago species reflected with the square lanceolata and the triangle Buguy. And you can see that it's still reflecting that pattern of microbial communities clustering by site type and being different between site types. So we see that the urban are clustering with each other, but there seems to be some separation once you go into the adding another layer of the Plantago species. Same here, sort of with the rural, and this is also again reflected in the fungal data as well. So, permanova results again showing that this interaction does seem to be significant. So going back to the predictions I had, right, of one, does the soil abiotic properties vary across level of urbanization? And two, do the soil microbial communities also vary with the level of urbanization? And I think so far the ordination plots are kind of saying, yes, these are supported. And now I'm going to actually transition into some of the taxonomic bar plots that I have of this data and some of the preliminary trends. So here we have the relative abundance of the top 10 bacterial phyla, and these are grouped by site type, as you can see by the bottom, rural, suburban, and urban, and these are actually just grouped as one. I haven't separated them out by Plantago species, just for simplicity's sake. Yes, and we see that the top 10 phyla, these are all soil and plant associated taxa. And I'm not going to go into too much detail for these bar plots, because I still need to statistically test if there are differences in the relative abundances of taxa, but I will highlight what I think are some visual trends. And hopefully you agree that these do seem like visual trends. So one of the visual trends for the bacterial data I'm going to highlight is that I'm going to actually use a black box to designate that trend, and I'm going to use the star next to the phyla to also designate the phyla of interest. And so we see that with the bacterial data in rural sites over here, that they have lower, I'm going to stumble over this, microbiota. They have lower of that phyla compared to suburban and urban. And so this is one of the examples of the taxonomic bar plot trends that I've seen visually, but still need to test for. Now I'm actually going to shift to just the fungal data now. So looking at the fungal data, this is again site types on the x-axis, rural, suburban, and urban. And these again aren't separated by Plantago species. And this is all 20 phyla that had been identified. And again, we see visually at least some patterns of different abundance differences between the site types, but again, they still need to be verified by statistics. And one of the examples I really want to highlight is that rural sites have higher glomerulomycota compared to the suburban and urban sites. And glomerulomycota, I think, is very interesting because that's the phyla that is comprised of orbiscular mycorrhizal fungi. And so these are symbionts in the roots of plants that help capture nutrients from the soil. And again, this is just one of the examples of the taxonomic bar plots for the fungal data. And so to kind of summarize the microbial data I've shown you so far, we saw that the microbial communities of both Plantago species overlap, with the Brugelii kind of being nested within the Lanceolata microbiome. We saw that the microbial communities also differ between site type, and that when you further separate them out by Plantago species, they still cluster by site type, but there's still a degree of separation between Plantago species. And then lastly, I've shown you some of the preliminary trends that I've noticed with the taxonomic bar plots for the fungal and bacterial data. But I still definitely need to further look into the taxonomic bar plots to look at a finer taxonomic resolution, at least to the genera level, and to verify exactly how different are these taxa across the different site types and across Plantago species. So recognize that, right, these are just trends so far, and there's still more that needs to be done, like always. And I'm interested in understanding how the soil chemistry and microbial data are linked at the sample level, both within and between sites, and whether or not certain chemical environments are selecting for certain taxa. So for instance, are sites that have higher heavy metals selecting for more heavy metal tolerant taxa? And I plan on using a Mantel test to test the correlation of the different chemical variables and the microbial taxa, not only at the site level, but at the sample level, to kind of get a finer spatial scale of these sites. I also want to look at, again, the abundance of taxa across the site types and across Plantago species, which I plan on doing a differential abundance analysis for. So zooming out of all this, what's the point of it all? So for plant communities, we can better understand how plants and their microbiomes respond to urbanization and heavy metals, and we can use that information when making decisions about urban planning, urban greening, and soil bioremediation projects. For instance, urban restoration projects that want to plant native vegetation would want to know the quality of the soil before they begin planting anything. Urban gardens might want to know if there are any pathogenic microbes in the soil before they, again, plant anything else. And that is the end of my presentation, and these couldn't have been done without the wonderful help of these people and my funding. Love money. And I'm just going to end it there. Still, I'll take questions now.
All right, I can just talk about that works. Great talk. I have a question about how you classify rural and urban. So was it based on management practices? Was it total population? Can you delve more into how you categorize and classify those two types of sites?
Yeah, so I would say like the main variable that we used to classify the different site types was by percent built environment. So that's buildings, roads, buildings and roads, I guess, the biggest things I can think of, sidewalks. And so 5 to 20% built, that's rural. 30 to 50% built, that's suburban, and anything above 50% built environment would be considered urban. But there are other variables that people could use to differentiate between these different site types. So this PCA, right, is showing like the other environmental variables that could be considered. I think different urban ecologists will classify it differently, but I think percent built environment's a good starting point.
Just a question on that intersection density is road intersections?
Yeah, intersections of roads, yes.
Very awesome. Thanks so much. One question just about interpreting the NDS plots. I think there was the measure of R squared. Yeah, guys just not familiar with this, but I'm wondering if that can also be interpreted as like analogous to the human portion of variation that is being explained for these. It seems like they're quite low. I'm just wondering if kind of across these plots, is there a large is there additional portions of variation that might kind of perhaps fall outside of the results shown in the points?
Yeah, so for those not familiar with R squared in this test, so R squared is the explained variance. So yeah, it is very low, right? For at least the Plantago right? Like was that 2% for bacterial and practically 2% for fungal communities. Repeat your question again.
Sorry, it's a vague question. It's kind of open-ended, but like, are there additional factors that might also explain additional portions of variation, given that the model that I'm assuming that this model, just the type of species explaining it's significant, right? But explaining a small portion of variation.
So you're asking like for this example in particular, like could there be something that I'm missing in this Plantago test that could be explaining more of the variation?
Sure, yeah.
I feel like for Plantago, I suppose like one thing I didn't control when I was sampling these plants is I didn't control distance from roads or distance really from other factors. I also didn't account for the surrounding plant communities either. So I could be missing some other background variation in this that could be explaining this trend, at least for Plantago.
I just follow up on that. One of the questions that I have is how much variability do you find within a single site, a single sampling site? I know that when you look at some of the other kinds of aspects of soil variation, you get a lot of variation within, let's say, in an acre, and so I think that might be contributing to some of the variance that you get. What is the within site variance? What is the variance among urban sites or rural sites? And I think there's probably an added component of variance due to that.
Yeah, so that actually reminds me of, there's a paper by Wu and Tonovics 1970s that looked at Plantago lanceolata, and they were looking at lead tolerance of that plant and controlling for their keeping consistent the distance from roads. So the closer you go to a road, the more lead you're going to have. And so those plants that are closer to the roads had higher lead tolerance. And so that is a factor of like distance from road or distance at a corner compared to like the more interior part of a park, for instance. I think someone had a question with the mic.
Hey Mahal, great talk. I just had a quick question about your sampling sites. For the rural selection sites, would there be any sort of difference since you're choosing protected areas such as Tyson Research Center and the Shaw Nature Reserve? Just because maybe they're not as like specifically a little bit more protected, maybe you're seeing some differences in soil composition or levels. I just wanted your thoughts on that.
Yeah, I guess it's like kind of the reason why we chose them as rural sites, right? They're very protected. There's not as much human involvement, for lack of a better word, in those. So as far as I know, there hasn't been a history of industrial waste at Shaw Nature Reserve. Oh, they do burn. This is true. But with heavy metals, at least, I don't know any knowledge of that. So you're asking like if I had gone to a less protected but more rural site, like someone's like farm, for instance, could there be a difference? Yeah, there definitely could, right? Farming sites have higher inputs of fertilizer and other pesticides that they could use. So that could be affecting the soil chemistry and the soil microbial communities as well. Yeah.
Thanks.
Hey, Mahal, thanks for that thought. I guess I had a question on what sort of ecological filtering is happening in some of the communities assembling these microbial communities. Has there been sort of ex situ experimental studies that are looking at how some of these different microbial lineages do in the face of these different heavy metals? And then have folks been able to compare that and how they're actually doing in nature when there's all these other sort of ecological covariates in the mix? That makes sense.
You're saying, or at least for the microbial communities?
Yeah.
Okay.
Yeah.
I want to say yes, because there was, this paper 2022 mycorrhizal infection can ameliorate or reduce abiotic factors in urban soils. So I believe in that study, they had like AMF inoculates, and then I believe put them in with plants with like in urban soils. And I think the plants did better than those that didn't have the AMF. So there probably is work ongoing. as to how like microbiomes that are maybe better adapted to a more urban environment can increase the health and fitness of plants in urban environments. There's also what I just mentioned previously, the Wu and Atonovics paper that was focusing more on like lead tolerance and seeing that plants that were already accustomed to lead tolerance, their seeds did better when they were exposed to higher lead as well. Yeah.
Oh, okay.
Continue.
So I just had a question about the suburban sites. Did you at all make note of like maybe the neighborhood age or things like that? Maybe if there might be some variance within the suburban site itself and lead content that could affect microbial communities.
So that's definitely other data that I did not readily account for. Right. Like the history of the park, how when it was first started and what's been the management history of the park since then, those are all factors that could be influencing the trends I see now. So if I have that data, not currently, no, but it is something to consider.
So I have a question that just basically about the soil. Did you take into account not just the location of the soil, but the nature, like clay, sand, organic? Because I imagine that influences soil chemistry a lot.
Yeah, I would say at least for the suburban and urban, there was pretty similar. Not sandy, not loamy, it was just organic matter. But I think at least when I'm remembering how the collections went, for Tyson Research Center, something that was very interesting was that I could only go to a certain depth. I couldn't go past like 2.5 centimeters, if I'm remembering correctly, because it could be the history of Tyson, because I believe it used to be like a military complex. Once you hit like past 2.5 centimeters, you're hitting like bedrock already, or like concrete or whatever it was that they have developed below. So you're right, like the types of soil that I encounter can really influence what I might see in soil chemistry and microbiota.
And then, well, I'll ask my other question after the review.
Okay.
All right.
Okay,
We'll
cool.
wrap up to the next topic.
Oh my God.
Yeah.
Yeah. But then I got this thing. Oh, when I was happy when I was like a 6 year
You know, and really look at the average age. So, you get the average age of houses built, you get a buffer. Every.
Thanks, Mahal, for that good talk. Obviously generating lots of conversation, which we're excited to continue after our next talk at the reception. So up next, we have Wenhsi Kuo, who graduated from undergrad from Taiwan University. And he arrived here in St. Louis in 2018 and is now a Scorpio in Ken Olson's lab. He did not give me a lot of information to go on, so we had to embellish. He is hoping to defend next semester, so stay tuned for a defense announcement, hopefully. But in the meantime, here's Wen.
Thank you for the cute intro, and I will provide more information. I like plants, raccoon, and goldfish, and I like to play badminton. And I'm now the 6th-year graduate student in Ken Olson's lab, and I have been working on clover for 5 and more years. And today I'm going to talk about my third chapter in my dissertation, and I'm still working on it, especially for the discussion part. And this is about temperature gradient as a major driver for population structure and environmental adaptation in North American white clover. So let's talk about environmental adaptation first. So like species are always trying to increase their fitness in their corresponding environment. One classic example is the paper moss in England. With the industrialization in the 19th century, the air pollution in the urban area they prefer the melanic mode of the paper moths because it can provide better camouflage against the predators. And with the nature migration between urban and rural environment, you can see there's a color climb from melanic black to white. And this conspicuous color pattern guided people into the early study of local adaptation. And here is another beautiful example. These are the sea snail in Spain. So we can see like the sea snail in the interior zone of the ocean. They are very light color. And when it gets to the exterior zone, the color becomes darker and then black, and black with white strap. And this color climb is consistent with several environmental gradients. For example, wave exposure, temperature, and salinity. So this factor could be the driver of selection. Although phenotype climb could be a result, of a genetic drift or gene flow. But if this climb pattern can stably persist for a long time, or maybe it can repeatedly occur in different geographic regions, it is pretty likely to be a sign of adaptation. While phenotypic climb, it is a very good approach. It is not perfect because it is only restricted to some simple and conspicuous traits, for example, like colors, and other important traits like fitness, because they are often in an intensive interaction with the environment. So they are not able to observe directly. For example, we can see the left plot. In a low elevation, the plants show a nice climb from high to low. But this climb pattern does not persist in the medium and high elevation. That is because the environmental interactions. So for most phenotype, a common garden or a controlled environment, it is still indispensable to document those traits. However, common garden experiment, it is expensive and laborious, and it can end up in some negative result, which is very disappointing to graduate students. Yeah, fortunately, there is a way to bypass phenotype and just to study the genetics under selections. Thanks to the advance of sequencing technology and globally available climate database, we are now able to investigate adaptive genetic without access to the phenotype. In this triangle, there are environment, phenotype, and genotype. And to investigate the relationship between environment and phenotype, this is reciprocal common garden experiment. And to investigate the relationship between genotype and phenotype, that is GWAS or QTL mapping. And for the relationship between environment and genotype, this is a relatively new approach. And what I am doing for this study, it's called landscape genomics. And here, I'm going to tell you why white clover is a great system to investigate this, to use this approach to do investigation. First, we have robust evidence of local adaptation in white clover. In the previous Ocean Lab, the previous Ocean Lab graduate student, Sarah Wright, she collected several white clover populations across the United States and planted them in Tyson Research Center. And she found a decline of fitness when the increase of distance to St. Louis. And the St. Louis local population had the highest fitness. And this home site advantages, it is a very strong support of local adaptation. Oh, I forgot the animation. Yeah. And in addition to this fact, white clover is there is a well-known local adaptation trait called cyanogenesis polymorphism. Some, but not all, white clover can actively synthesize and release cyanide when herbivore rupture the cell. So they are poisonous, and the other, they are innocent, they are edible. And those poisonous clover, they are mainly distributed in the south. On the contrary, those edible clover mainly distributed in the north. Some hypotheses state that because the south side with warm climate, they have more herbivore pressure, and maybe it's more dry, and the dry may associate with this kind of poisonous trait, but it is not sure. And in the north, the climate is colder, and which have lower herbivore pressure. And also, physiologically, the cold temperature can constrain the cyanide biosynthesis. And also, in the north, when the frost can cause leaf freezing, and the leaf freezing can, the water molecule can rupture the cell, and it can cause ototoxicity if the clover is poisonous. And we also know the genetic mechanism of cyanogenesis polymorphism. It is controlled by two unlinked genetic loci. The first loci is AC. AC can convert an amino acid to cyanogenic glucoside. And LI encoder protein can hydrolyze cyanogenic glucoside to release toxic cyanide. So they are full functional combination. Only the plants with functional AC and LI loci, they are poisonous. We call them cyanogenic. And the other three combination, they either have only one functional unit or have nothing. They are not cyanogenic. And from our very recent study, we know this kind of loss of function allele for either AC or LI, it is a complete gene deletion. And for those functional allele, it is a dosage-dependent copy number variation. With more gene copy, the plant can synthesize more toxic cyanide. So let's go back to this triangle. So here, the transplant and common garden experiment investigated the relationship between environment and phenotype. And the cyanogenesis polymorphism in clover, it bridged across environment and phenotype, and also genotype. So the still unexplored relationship here is between environment and genotype, and that's what I am doing in this study. And one good thing is like the result between environment and genotype can be compared to our previous finding. So to conduct landscape genomic study, we did the most comprehensive sampling to date in North America, including 415 individuals from 43 populations. So first, we want to investigate the consistency to the previous study. Is there a cyanogenesis climb across North American white clover population, just as previous study did? And this is our sampling site, and the different color in the pie chart show 4 different functional phenotype of cyanogenesis polymorphism. So only this green color phenotype can synthesize and release cyanide. So we can see they are mainly restricted to the very south population. And we find it is also quantitative. So in a warmer temperature, there is more AC and LI gene copy. So yes, it is consistent to the previous finding for cyanogenesis clone. And then we want to ask, like, because like we know, the functional AC and functional LI phenotype, we call big AC and big LI phenotype, it can be selected for its defense ability. And the small AC, small LI phenotype, it can be selected for its energy efficiency. But what about the other two? They can only synthesize one functional component. So is it just a waste of energy? If this hypothesis is true, we would expect to see the lower number of counts in these two phenotype. And we find, yes, the small AC big LI and big AC small LI phenotype, they have a lower number of counts compared to the Hardy-Weinberg equilibrium. So, so far we use cyanogenesis cline to build a connection between environment and phenotype. And then we want to make it comparable to landscape genomics. We still need to bridge this gap, the phenotype and genotype. So that is GWAS. We use the same data set and we successfully detect strong genetic signals for the AC and LI phenotype. And the location are consistent with our previous knowledge. So until here, we are almost ready to do some landscape genomic study, except that we need to know the population structure of white clover in North America, because population structure could be an important confounding factor for landscape genomic analysis. So this is the mixture output. The x-axis is the 43 population based on the sampling site. We can see color as the genetic composition of that sampling population. So we can see this color pattern, they are not random. So yes, there is population structure. And if we arrange the population by their latitudinal location, we can see a nice pattern from south to north. We also find the 5 southmost population. They look distinct when the ancestral population number set from K to 5. They somehow stand out. It can be better visualized on the map. As you can see, the south population, they seems to have a different genetic composition than the other North American population. So since there is population structure, we want to see if the population structure can be better explained by climate than geographical distance. In that way, the population are more likely to be under environmental selection. So this is isolation by distance. The x-axis is the geographical distance, and the y-axis is the amount of genetic differentiation. So we can use this as a non-hypothesis. So if there is some selection, we will expect to see more correlation between climate and genetic differentiation. And that's what we see here. So among a list of climate variables, we find varying degree of 5, which is a temperature gradient, show very nice association to the genetic differentiation. So this is a very supportive evidence to show maybe there is some selection going on. So finally, with the control for population structure, we can conduct landscape genomic to investigate if there is any gene that stand out. They have extra or additional association to the climate variable. Because for those genes with additional extra association to the climate variable, it is pretty likely those genes are under direct selection. And also, like we expect to see the cynogenesis genes, the AC and LI, they should be included in those association outlier. And here is the result. Yes, we find there are multiple genes related to flowering and developmental regulations. So these are the genes, and the big F later means it's a flowering regulation related, and the D means it is developmental regulation related. So there are several genes related to those dysfunction in the association outliers. However, we didn't find the cynogenesis genes in those significant collection of association. So the AC genes, there is a peak there. So it means there is a signal, but it didn't get above the threshold. And for LI genes, it is no signal. So from the result, we have some new hypotheses. So the new hypothesis is like the variety history divergence rather than cynogenesis polymorphism could be a more important fact under the local selection in white clover. So this is my hypothesis. So because like in the south side, the temperature is pretty warm in the spring and hot and too dry in the summer. So the clover layer, they need to finish their life cycle early. But for the nose population, because the spring is still too cold and the summer temperature is decent, so they can just keep doing more vegetative growth in the spring and flower later in the summer. So from the result of population structure and also those developmental genes, flowering regulated genes from the landscape genomics, I hypothesis because of the divergence of the reproductive time, reproductive schedule, there is a barrier for gene flow between the north and south populations. And cynogenesis, although it is still an important trait in local adaptation, its role in the overall fitness of adaptation still need some further investigation. And yeah, that's it. And I need to thank to my advisor and our LabTech and their members and also my founding souls. So any questions?
That's a very interesting talk and a beautiful system. I have one suggestion based on some of my own experience. You notice that the disequilibrium between the two genes you know are responsible for gynogenesis, or it didn't show up on that G plus type thing. Yeah. And a former graduate student here, Charlotte Clymer, came up with a very different kind of measure. The problem with these things is they're all scalars. You have one number associated with all those dots, and she had a vector way of looking at associations between genes. So you can look at specific allele combinations, which is what you're interested in. We used it to look at the skin color genes in humans, and using her measure, her vector correlation, We got beautiful clients with ultraviolet radiation that weren't obvious with ordinary measures of this equilibrium for association. So you might want to consider using Charlie's approach as well.
What do you mean the vector approach for
Okay.
allele?
Like a linkage disequilibrium statistic, what you get, you calculate, you get one number.
Yes,
But suppose you have two loci and two alleles.
yes,
You have four combinations. The vector gives a specific number for each of the four combinations. So you don't have one number, you have four numbers. Much more powerful for looking at associations between specific alleles. You throw away a lot of information by putting it into a scalar. And you can retrieve that information by looking at the specific combinations that are important for local adaptation. I can give you a reference.
okay, that would be great.
Yeah.
Yeah, thank you.
Sure.
Anyway, nice talk. Is there any sort of geographic area with these rivers down in southern US at all, or these floaters sort of dispersed across them with no problem?
I am not sure. And like for example, Mississippi River, and I think like because our sample was not dense enough to investigate the barrier, the effect of a big river in geography. But I think it is likely because clover seeds, it's the seed size is not small. So I think like the seeds have limited dispersibility. Seems like they are flowering plant and they are out closer. So the pollen, it's a wind dispersed pollen, or something like it's like insect pollen, insect dispersed pollen. So like maybe it can disperse across rivers. I'm not sure. Maybe more dense sampling is needed to answer this question.
What's happening in SCS or SCT? What's happening with that site?
Oh, so SCT is yeah, it looks very different.
Yeah.
So also in the structure result, you can see FCT. They have a very different color compared to all our populations. However, I have no idea. It's not from a single individual. It's an average value of 10 individuals. So the 10 individual from Connecticut looks different. I think like if I have an opportunity in the future, I like to go back there and to do some new sampling. Maybe at a time we did sampling, we get some buyers collection in that site.
It might be somebody's garden, so it could have come from who knows where.
Yeah, because like people, I think people like intentionally plant clover for nitrogen fixation. So it's, I feel like I didn't do it myself, but I believe the sampling of clover could be tricky because we want to sample some naturally adapted, naturalized clover, not somebody just planted it.
Yeah, I think we're invited.
So I was just following up on that. I was also wondering about it possibly being moved around by vegetative spread. And then also, is it part of hay? So if somebody pays a pasture and then sells the hay, you could have a lot more dispersal, and that would be with seeds and flowers and things.
Like mixture in a haze and
Yeah, and then it goes to another farm, and if it's a horse pasture, they'll throw the hay down, and we end up getting clover.
yeah, I'm thinking it will be very interesting if I can have both European and American samples, and so that we can investigate the question: do the selection work on standing variation, or do the selection work on new mutations? And that seems like we now like European samples. So yeah, we have limited explanation here. Yeah. Hey, Justin.
I have one question for. One question for your final, plot where you had the map and drew the line that separates the southern from the northern populations. Yeah, thanks. So just wondering about how big the difference in flowering times is expected to be to separate the southernmost from the middle populations. Because I would imagine that the difference in flowering times between the middle and the northernmost populations could also be equally as large. And so I'm just wondering if you had to draw another line across this map, where would you place it?
I think this question can be, is the flowering time differentiation continuous? Or maybe that is discrete. And so far, I don't know. We have sampled 3 population. So the left panel is population from Minnesota views and Missouri St. Louis and Florida Gainesville. So the upper panel is the fluorescence count after the day of planting. So you can see the plant from very south population. They just want to flower soon, early than the St. Louis and the Minnesota population. We just have 3 population here. So I think we do need some more comprehensive investigation. And I think like a temperature, especially like sorry. This is a growing degree day of flight. So this is a product of temperature in days, and it is above 5 Celsius degree. And farmer use this one to estimate when or how many crops they can plant in a year. So I think temperature, my hypothesis is it is not continuous or linear relationship. It may get above some temperature threshold and the plant can flower. But that's just my hypothesis.
Thanks so much. That's such a great answer. I also just want to end with a comment that I wonder if there might be an opportunity to connect this study with the asynchrony of seasons hypothesis, which is essentially an interspecific interpretation of some of the physiological differences that we're talking about seeing here within species. So,
like compared to white clover relatives,
for example, but we talk about that later.
Yeah, I'm thinking like because white clover is an introduced species, and if we can find a native trifolium, the same genus species with wide distribution, maybe it will be a good comparison. And yeah, thanks for the idea.
Thank you all very much. With that,